All right, good evening. I'd like to welcome you all to the 16th annual Tyler Darwin Day. Before we get started, I just want to thank a number of people. First of all, Danielle Pritchard is the co-chair for our committee, and this is her last year. So can we give Danielle a little round of applause? Danielle is a major help and makes sure that this event goes off without a hitch every year. And so we will definitely miss her as she goes off to better things. And then the rest of the Darwin Day committee has been very nice to us and helped plan events. We've had a lot of help from our Tri Beta Honor Society as we've had several events this week. We went down to the Discovery Science Place with Tri Beta, with the Wilderness Society, and with the Social Sciences Club. And we had a great time. We were joined by Texas A&M Overton's Extension, and we had a lot of great exhibits for the children. So I thank the students that came out and helped with that. On Tuesday, we had our UC information booths, and again, Tri Beta, the Wilderness Club, and the Social Sciences Club all helped out with that. On Wednesday, we had Tri Beta helping out with the movie nights here on campus, and the Earth and Space Science Center, who are absent tonight because they are running their big Love Under the Stars event, but they helped plan our planetarium movie as well. And then we can thank the STEM Club for hosting the lunch yesterday. And I'd like to thank UT Health Science Center for hosting the lunch today. Tonight, we're very fortunate to welcome a speaker from the University of Colorado Medical School. It is Dr. James DeGregori, and he is going to be talking about how evolution impacts cells in the body as they develop. Some of it may be based on the book he has written called Adaptive Oncogenesis, A New Understanding of Cancer as it Evolves Inside of Us. He is currently the Courtenay C. and Lucy Patten Davis Endowed Chair of Lung Cancer Research and the director for the University of Colorado Cancer Center. He has over 80 published articles and book chapters. And his lab focuses mostly on cell culture and mouse models of cancers, including leukemia and lung cancer. He started off his career as a Bachelor of Arts microbiology major down at UT Austin. So he is from Texas and, well, from Ohio, then Texas, right? Something like that. And we're very fortunate that he's able to come back and talk with us. He got his PhD from MIT in Boston, and then his postdoc was at Duke University. Currently, he is the associate editor of Evolution, Medicine, and Public Health, and the editor-chief of Aging and Cancer. And one of the things I really hope you can see as he talks tonight is that evolution does play a major role in our practice of medicine and how we should think about disease and cancer. And he is the holder of 2 patents that are both focused on technologies involving cancer and treatment of cancer. So without further ado, I'd like to welcome Dr. James DeGregori, and he will tell us about
Now it should be on. Yes. So it's a real honor to be here. I've never been to Tyler before, even though I grew up in Texas. So thank you. All right. So we're going to start off with, a simple kind of question. Why do we get cancer and other diseases mostly at older ages? And this is a problem that my lab has been, sort of focused on for probably a good quarter of a century right now. And one of the sort of modeling studies that we did that I think shed a lot of light on this was this one. And I could go through a whole lecture just on telling you why we need to think beyond simple accumulation of mutations. But I'm just going to give you this one piece of evidence to sort of suggest that there's something much bigger than the simple accumulation of mutations that's at play here. Let me see, is this a pointer? Yeah. So if we think about different cancers, these are leukemias, and they have different curves of incidence, with, for example, CML is less common than CLL. And they're initiated by different numbers of driver mutations. Like CML is pretty much known to be, when most diagnoses, it's just a single mutation that drives it. And then you have different solid cancers over here, like colorectal, that's thought to have anywhere from 4 to 11 different driver mutations, depending on how you define driver. And these different cancers, like the colon cancers, arise in these isolated stem cell pools in your guts, where you've got a dozen or so stem cells across about 10 million different niches. So on the order of 100 million different stem cells. In contrast, the leukemias are thought to initiate within the bone marrow and our hematopoietic stem cells, and you have roughly 200,000 of those. So many orders of magnitude difference in the stem cell pools, probably about an order of magnitude difference in the number of mutations required to get these. And so you think this should lead to very different curves. If these are just limited by those mutations, they shouldn't all look the same. And yet, when we do a very simple thing, we being Andre Rosak, where you just basically ask, if you just normalize all these to their maximums, they fall right on top of each other. And so I think this indicates something bigger than simply mutation accumulation. And I'll get into what that is as we move along. And so when Andre did Monte Carlo modeling, which is a computational modeling method, and he asked, well, how can we model these differences in cancer incidence with a common overall shape of this curve, with the knowing that it requires different numbers of mutations and very different stem cell pools where they're initiating. And he found that he needed a factor where you had negative selection against the mutations in youth, and selection for these mutations at older ages. That factor was really required to allow this modeling to happen. And so essentially the way we can think about this is what this really reflects. This is the force of natural selection pushing these diseases off to the right, meaning off to older ages, typically beyond 50. And again, I'll get back to why I think that is. So you might think, well, how is this accomplished? How do we avoid cancer? We've got 5 to 7 trillion nucleated cells in our body as an adult. So how do we keep those cells in line, keep any potential malignancy in check for a good half of a century? And I think that's where we need all of these different tumor suppressive mechanisms. And I'm not going to go through all these, but it's everything from maintaining your genome, preventing mutations, preventing epigenetic change, through getting rid of cells that are misbehaving, through competition, through telomeres, and controlling inflammation, preventing metastasis. But what I think is important here is we have to keep in mind, our bodies are laboratories of somatic evolution. There is a ton of somatic evolution happening in our bodies. And yet cancer is still rare from the perspective of, out of every 5 to 7 trillion cells in an individual, one of those, and about 40% of us, will become a cancer. And it typically happens late in life. And while that's tragic when it happens, we have to recognize that that's the rare case of a cell actually making it through all these different steps. So in the vast majority of cases, these barriers are effective, and they prevent the cancer from actually killing us. At least they prevent it to the point where, at later stages of our life, beyond 50, for example. But what I'll also note, you see that these boxes become sort of, basically dotted at these later points. And that's to indicate that as we get old, it's not that these mechanisms go away, they just become less effective. And it's one of the reasons we think that we start to get more and more of these cancers at later ages. And so I think the 10,000-foot reason, the evolutionary reason why this is the case, is that we were very unlikely to be alive at ages where most cancers are currently happening. So this is the historical survival of humans, and this is taken from curves that are very similar in Africa, in the Americas, and Europe, but I'm talking 1,000+ years ago. So at points where most humans were dead by the time they were 50. In fact, many humans didn't even make it to the age of 5 because early childhood mortality was so high. And if we think about modern rates of cancer, and really this could be modern rates of heart disease or many other diseases of old age. It could even be death by COVID. They're mostly happening at the second half of a century. And we believe that this is because basically there's been investment in our soma, our bodies, to the extent that maximized reproductive success. And so this investment will start to wane at older ages. So it's not like there's a program to get rid of us. Cancer isn't a program to weed out the old to make room for the young, but instead it's the waning of programs to maintain us and to prevent such diseases. And that is basically has an evolutionary explanation, and that's that basically natural selection does not act to keep us alive forever. It only acts to maximize our reproductive success. And by us, I mean all animals. So it's an example. I'm going to use clonal metapoiesis. And I'll get into what this is in a second. But clonal metapoiesis are these clones that are detectable in the blood of individuals, and they become much more prevalent in older people. So 15 to 25% of people over 65 or 70 will have these clones. They're often driven by mutations in genes that are known to be mutated in leukemias. And yet it's not really a leukemic condition, but it does have health consequences. So you can see that the prevalence is very low, pretty much 0 up until 40, and then starts to rise exponentially at older ages. And this is often driven by, for example, these are some of the top genes where mutations in these genes are driving this clonal metapoiesis. And these same mutations can be found in leukemias, but not always. Sometimes it's actually the same, the gene may have the mutations that are common in leukemia, but many mutations that are found in clonal metapoiesis are not actually the ones that are actually found in the leukemia. And these different mutations, different genes have variably are associated with age, but all of them have some associated with age, meaning these expansions become more prevalent as the people get older. Some of them can be associated with therapy, probably leading to their selection, even smoking. But what's also interesting is they're variably associated with the risk of malignancy. And what I want to point out is 3 of them, because we'll actually be studying them here. DNMT3A has really a very small increased risk of malignancy. Keep in mind that this line here at 1 would indicate no increase in risk. So anything to the right of 1 indicates an increased risk. So for example, just to be simplistic, a hazard ratio of 2 would mean you're basically doubling your risk of malignancy if you've got one of these expansions. But you can see TET2 is a little bit higher, and UTF1, which is a splicing factor, is even much higher. It's a 7 or so fold increase in the risk of this malignancy. So different risk for different mutations. And what's more is this isn't just about leukemia. In fact, if you've got clonal metapoiesis, or what we call CHIP, you have an increased risk of dying. Most of that increased risk is not due to leukemia. It's due to cardiovascular disease, such as heart failure. And there are many other diseases of aging, even gout, which I kind of find interesting, that are associated with this clonal metapoiesis. So it's highly associated with diseases of aging in general, not just leukemia. And the other thing that's interesting is if you compare, for example, peripheral artery disease, or really, sort of a cardiovascular disease, to hematologic malignancies, their increased risk for each of these are relatively in line with each other. It's sort of a very close association. So a mutation like DNMT3A that is very unlikely to increase the risk of leukemia is also very unlikely to increase the risk of peripheral heart disease, but splicing factor mutations like the U2AF1 increase the risk of both much more. And this is going to be relevant because I think this is essentially their fitness effect on the organism. It's not just through leukemia, it's through general effects on your ability to survive if you have these expansions. So there's been a number of reports that have shown that clonal hematopoiesis, this process that I'm talking about, the IP is just for of indeterminate potential, but let's just call it clonal hematopoiesis to keep things simple. It's associated with aging, which I already showed you, but also with inflammation. And I'm not going to go through all this work, but just to put that out there, that it's known to be associated with inflammatory conditions. And if we think about this association, I think there's two probable explanations. First off, your stem cells, HSCs, hematopoietic stem cells, your blood stem cells, basically, as you get older, they're exposed to sort of damage and epigenetic drift that would happen just over the process of time. And this is sort of ratcheting down their functionality, their fitness. At the same time, we're always getting exposed to different infections and, everything from viral infections, even dysbiosis in our guts. And this is also probably contributing to sort of this reduction in the competency of your stem cells. And we know that this becomes hardwired, meaning you could even take these old stem cells, put them into a young mouse, and they still act old. They still have the features of old stem cells. So some of it becomes embedded in those cells. At the same time, we know that aging of this population and the poor function of this population is also due to the niche. So, your bone marrow becomes more inflammatory, becomes more fatty in old age, and that is not as conducive to a happy, healthy young stem cell. So now we're going to ask the question, is there selection for these CHIP mutations in old age? Are they being differentially selected? Remember, the modeling I showed you before suggested that in order to get these aging curves of, increased aging incidence for leukemias and other diseases, we really need a differential selection, like reduced selection or even negative selection in young age, but positive selection in old age. And so we asked the question, is this the case for these different mutations? And this is Marco De Dominici, a postdoctoral fellow in my lab. And so Marco developed just really a fantastic protocol where he uses CRISPR as a means to induce these mutations. And he's inducing CRISPR in these hematopoietic stem cells. And I won't go into the details, but he can do this with very high efficiency. And we also use a conditioning method that's very mild. A lot of studies have been used a radiation to condition the host to be able to put the cells in. The problem is the radiation itself is a really strong perturber of the tissue environments. So we wanted to avoid that. So we came up with a way of sort of just tickling the environment just enough to get the cells in there, but not much more than that. So we can get these cells in, we can efficiently induce the deletions in them, and we're purposely getting them in where only a few % of the cells will have the mutation, so that they're largely surrounded by wild type or competent or competition. And when we did this with DNMT3A, which is first project, he got the surprising and what was then very disappointing to him result. But as I've often said to students, you shouldn't be too married to your hypothesis, because when your hypothesis turns out to be wrong, that may be a blessing in disguise. And in this case, I think it really was. So what he found is that we got sort of a modest selection for DNMT3A mutations that was very similar in both young and old hematopoietic systems. So there was no aging dependence, which is surprising because even our modeling had said there should be aging dependence. And we see the same thing in the bone marrow, no difference in the stem cell pools. But then he turned to TET2, this other mutation. And remember, this one was a little bit more dangerous than DNMT3A in terms of its effect on your survival, right? Your peripheral artery disease or your leukemia incidence. And then he found that indeed now we see very clear that there's better selection for TET2 mutation in old stem cell pools than in the young stem cell pool. This is looking at peripheral blood. We see the same thing when we look in the bone marrow at all different sort of stem and progenitor cell populations. But really strikingly, when we look at the hematopoietic stem cells, the ultimate source for all blood generation, we see much stronger selection in the old stem cell pool and essentially no selection for TET2 in the young stem cell pool, keeping in mind that 1 basically means neutrality. So there's no selection if it's at 1. So this would suggest that if you got a mutation in TET2 in your stem cell pool when you're young, it will not be selected for, but it would become selected for when you get old. And so then he wanted to ask, well, what's the mechanism underlying this selection? Why is it aging dependent? So he did a competition experiment where he co-transplanted knockout cells with different competitors to see which was sort of the more fit. This is basically a classic fitness test because the ability to sort of propagate your type is a measure of fitness. And when he puts these old TET2 knockout cells against old wild type cells, they win hands down. And I'll note that again, 1 is neutrality. So this is meaning, for example, at this last point, these have over 6 or 7-fold increased advantage over the control wild type cells. And if he puts the TET2 knockout, old TET2 knockout cells against young wild type cells, they actually have a pretty minimal advantage. It's actually close to neutrality. They still have a little bit of an advantage, but they basically, the wild type cells are much better at outcompeting these than the old wild type cells. But interestingly, the old TET2 knockout cells are actually have a lower fitness relative to the young TET2 knockout cells. So this indicates that it's not that the old cell is somehow primed to be a better cell when it's TET2 knockout. It's just that this old cell with the TET2 knockout is competing against a bunch of crappy old wild type cells, and that gives it its big advantage. But the young TET2 knockout cell would actually be a better cell, a more fit cell. And finally, old wild type cells have a much lower fitness than young wild type cells. And that's just consistent with data that's been out there for more than 20 years that stem cells become less functional in old age, less fit. So basically, the key point is that the fitness differential is greater between old TET2 knockouts and old wild type cells than the young TET2 knockouts and the young wild type. And in fact, if we really look in the stem cell compartment, as I showed you, the young TET2 knockout have no advantage over the young wild type cells in an actual stem cell compartment. So the next question is, how do we sort of get an understanding of what is TET2 doing in these stem cells, TET2 knockouts, the TET2 disruption, in order to provide this fitness advantage to the cells, specifically at older ages? Why is it adaptive in the old stem cell pool and not the young stem cell pool? And so to do this, we did single-cell RNA-seq, which was sorted from young and old stem cell pools with and without the TET2 knockout mutations. And we're comparing it with the wild-type cells in the same host mice. And this is a very close collaboration with Lamis Nadaf, who's a postdoctoral fellow in Shang Li's lab. And we've been collaborating extensively with Shang Li on a number of projects. And so when we did this, one of the things that they noticed was that we see this increase in the expression of a lot of ribosomal protein genes that is going up even by middle age compared to the young. So red is meaning more intense expression of these genes. But what's really striking is that in the middle-aged and old TET2 knockouts, specifically in the stem cell population, we see reduction in these genes. So it's basically reversing this ectopically overexpressed ribosomal protein genes. And you might think, well, why would expressing more ribosomal protein genes be something that would reduce the fitness in the case of the old stem cells, but have it restored by the loss of TET2? And I'll get back to that in a second. But what's also worth pointing out is that when we look at basically all genes upregulated with aging, they're pretty much all downregulated by TET2 knockout. So TET2 knockout is really reversing an entire program of gene expression that is ectopically increased as these cells get older, essentially rejuvenating them. Now, if it was just rejuvenating them, that would be a good thing for us, but it's not. As I said, this is associated with increased disease risk. So in addition to this rejuvenation of all these genes, we also get other genes that are being expressed that could probably contribute to pathology. And I'm not going to get into those today, but just to know, it's the genetic program that's being instituted here that's resulting in adaptation, selection for it, does not need to be the same program that contributes to sort of the malignant phenotypes that get conferred. So when we do analyses, bioinformatic analyses of these data, we can ask, well, what pathways are being upregulated? We can see that two of the top pathways that are upregulated in old stem cell pools are these RUNX1 and PPD3 driven pathways. And those get uniformly downregulated by the loss of TET2 relative to wild type. And this was really interesting because previous data from Nancy Speck's lab had shown that RUNX1 can regulate a large number of ribosomal protein genes. All these RPLs and RPSs, they're all components of the ribosome. And this is part of a stress response pathway. And losing RUNX1 would confer resistance to the stress by downregulating these same genes. So we thought maybe the same thing is going on with aging. And another important factor is another group, the Suzuki lab, had shown that RUNX1 and TET2 are co-localized at many sites of DNA regulating RUNX1 targets. So this basically provides a direct link between TET2 associated with RUNX1 and the regulation of all of these ribosomal protein genes. And so what we think is happening is TET2 regulation of RUNX1 targets leads to ectopic production of these ribosomal protein genes. And I'll mention it's also known that ribosomal protein genes, if they don't associate with the ribosome, which we think when there's excess production, they won't be able to. I'll also note the actual levels of the ribosomal RNAs that scaffold all these proteins. Those go down in old age in these same stem cells. We've shown that. And so you're going to end up with excess amounts of these proteins. This induces a stress response, and by binding to MDM2, which normally mediates the degradation of this key transcription factor, PPD3, that mediates stress-type responses, it's going to prevent MDM2 from binding to PPD3, releasing PPD3 activity, and explaining why we also see that upregulation of PPD3 transcriptional factors. And in fact, when we've looked at human data now, I think it's always important to take things from your model organism and ask, are we really seeing the same thing in humans? And the answer is yes. When we look at humans, so at these same pathways, these RUNX1 dependent and ribosomal protein gene dependent pathways, they're going up, which is denoted by the red, in these comparisons between either middle-aged humans and young humans and old humans and young humans. We're seeing this increase. And we see the same thing in particular stem cells in other data sets. But what's important is when you look in people that have clonal hematopoiesis driven by TET2, we see reductions now shown in blue in these same pathways. So the human data perfectly mirrors the mouse data. It's also important to note that PPD3 is known to be upregulated in many different data sets where they look at the stem cells with old age. So this upregulation of these PPD3 pathways is also really common in old age in human stem cells. Note that it's in the TET2 versus wild type studies that there were two groups of HSCs, and the group of HSCs that's known to be non-inflammatory is the only one that does not show this upregulation. I should also mention this is a collaboration with Angelica, who's in Stephanie Z's lab at Princess Margaret in Toronto. So what I've shown you so far is that old age will select for the loss of TET2 in hematopoietic stem and progenitor cells. And this could help explain why these mutations, these disruptive mutations, are found more in older people during clonal hematopoiesis. And the expansion of these TET2 knockout cells in old mice is due to the reduced fitness of old wild-type cells. The ribosomal, RUNX1 target, and PP3 target genes are all increased in old HSCs, but they're brought back down by TET2 loss. And so from data that I haven't shown you, we've shown that loss of RUNX1 or PP3 genes restores the fitness of these old hematopoietic stem and progenitor cells, suppressing TET2 mutant clonal expansions. I'll mention that this has been submitted for publication, but it's also out on a preprint if anyone wants to check it out. And you can also just Google my name. It's a unique enough name that it's easy to find almost anything. So Let me step back a little bit. I'm craning my neck back here. Maybe I'll just come down here. And so I think what's important to point out is that what I'm showing you is that what seems like a cell intrinsic effect of the TET2 knockout on HSCs is actually probably due to sort of a lifelong damage accumulation within the HSCs. So all this exposure during our life, the inflammatory cues, the niche deterioration, the epigenetic drift, is leading to this ratcheting down of the fitness of the stem cell pool, making it susceptible to this TET2 mediated clonal expansion. And I'm going to get back to this in a little bit because we're going to do some single cell studies to ask how do these changes in life change the ability of these stem cells to adapt to their environment. And this leads to this fitness differential that can allow for the selection of the stem cell, of TET2 mutations within the stem cell pool. So now we're going to turn to that third mutation. If you remember, the third mutation was in the splicing factor, U2AF1. And these were the mutations that conferred the highest risk of leukemia, but also conferred the higher risk of, for example, different cardiovascular diseases. So they're going to be the worst mutations that you would want to have expanded within your hematopoietic stem cell pool. And this is work from Bridget Ho. She's a graduate student in my lab. And the mutation that we're going to look at is this S34F mutation. And she's got a ton of data that I'm not going to show you for the sake of time, but I'm going to show you some really kind of like key pieces of data. So she did experiments where she took this mutation from either young or old mice, showing the old ones as gray, and transplanted these into young and old mice, but also did the cross transplant where we put like young into old, old into young. And when she puts these into the young mice, she got a really surprising result. They were strongly selected against. You might think, wait a minute, these are known to be in leukemias. They're known to be in clonal monopoiesis. Why would they be selected against? They're supposed to be transforming. But they were selected against. And note this is a log2 scale. So this is a big selection, a large selection against these cells. They're basically disappearing from the organism. But when she did the experiments where she looked at different ages, note that in old cells that go into old mice, she actually gets positive selection for the mutation. It's advantageous. So the very same mutations that are disadvantageous in youth become advantageous in old age. And I find this really fascinating. But what's also interesting is when she does the in-between, like old cells into a young recipient mouse or young cells into an old recipient mouse, she gets kind of in between. Maybe a little bit of modest selection against these cells with the mutations, but not a lot. It becomes much closer to neutral. This indicates that some of this differential selection is due to the age of the stem cell, and some of it is due to the age of the niche. It's kind of a combination of the two. And so we wanted to get to the mechanism, and the method that's best at doing this is single-cell RNA-seq, because it gives you sort of a phenotypic profiling of every cell that you can look at within the hematopoietic system, and you can compare the ones that do have the mutation to the ones that do not have the mutation. And we did this, and this is the stem cell population, and that's the only data that I'll show you is just on that one population, because it's really all we have time for. But we are looking at the effects on these other more mature populations. But when we did this, we got really kind of strong data to indicate why this mutation is probably selected for in these old stem cell pools. So we find that the interferon pathways are increased in old stem cells. I'm not showing you the comparison with young, but these go up in the old stem cells. And loss of sort of the mutation of this U2AF1 is reducing this pathway, shifting it to the left. This is showing it by another ways, but it's a very strong reduction in these pathways. And this is interesting because interferon stimulation has been shown to reduce the fitness of hematopoietic stem cells. So being resistant to this would be basically increase the fitness of a stem cell. In other pathways that are involved in cell growth, like EGFR and OXPHOS pathways, they're increased in the old stem cells that have this mutation relative to wild type. So things that might positively, promote the ability of these cells to compete within the stem cell niche are now being increased by the mutation. But you might think, well, why? But then that doesn't help explain why it's disadvantageous in youth. But we have data for that. In the young background, now these same pro-proliferation pathways are reduced in the stem cells. And this is true for this PI3 kinase pathway as well. So we're getting reductions in this pathway in the young stem cell pool by the same mutation that's increasing fitness pathways in the old pool. And actually you get an increase in apoptosis and stress response pathways in these young stem cell pools as well. So more bad stuff, less good stuff in the young stem cells, and the opposite in the old stem cells. So basically, to sum this up, U2F1 mutation is differentially adaptive in young and old stem cell pools. This S34F mutation restores more youthful gene expression in interferon, proliferation, growth signaling, and metabolism in the old HSCs, but it reduces gene expression and growth signaling and increases apoptosis and stress response gene expression in the young HSCs. Opposite effects in the two different age pools. So what we're doing now is we're analyzing actual splicing changes. This is much more difficult because we don't have a lot of these stem cells, so it makes the methods much more difficult. But we want to connect how is it that this mutation, which is in the splicing factor, is altering splicing factors to have such divergent effects dependent on the age of the stem cells and the age of the recipient mouse. So I think this harkens back to what I was telling you about before, this modeling that was done by a former postdoc in my lab, Andrei Rosak. And what he proposed in the same paper, which was actually kind of surprising because he stuck this model in the discussion, and actually our modeling hadn't really shown what he's showing here, and somehow the reviewers didn't catch that and let us put it in there anyway. But what he was basically proposing is that if you had a mutation, and that mutation caused a somatic clonal expansion early in the life of this animal, this make-believe animal, and if that mutation basically led to early death from cancer or anything else for that animal, versus other mutations like this blue and yellow ones that had a much longer time before they would lead to the death of the animal, they're less pathogenic. The natural selection would act differentially on these expansions. So the red mutation, natural selection, would push it down and say, hell no, you're not expanding in youth, because if any animal that allowed that expansion would be unlikely to be reproductively successful and be weeded out from the population. So it'd be strong selection for mechanisms to prevent the red mutation from causing an expansion. That's like the U2AF1 mutations. And in contrast, you will still get selection against these expansions from the blue and yellow mutations, but it just won't be as strong. So in the end, you get this equalization. And then we think this can help explain this sort of common cancer curves for these different cancers driven by such different mutations in different tissues. Basically, natural selection is tuned to each one to do just a good enough job to keep them from coming up during the period when these animals would likely be reproductively successful. So we think this is relevant not just to humans, but to basically all animals. And it's interesting if you think about this, because if you think about just these three mutations, their frequency is inversely proportional to the risk of disease. So DNMT3A, which is the one that causes the least risk of disease, is the one that's most common among clonal hematopoiesis. But U2AF1, the one that is associated with the most disease, is the least common one. And we think that's because basically what we've shown, and I've just shown you, is that there's the relationship is much more towards U2F1 in terms of the amount of negative selection in use, how much it's pushed down on, like the red arrow shown here. So natural selection has acted to maximize the fitness of the organism by sculpting a soma that selects against mutations to the extent that they would otherwise reduce our fitness, to put it simply. All right. And I'm going to stop and just take a question or two. I've got a little bit left after this, but I just wanted to, anyone have a quick question? Yeah. There's a little that I don't think for most of them that they're significant, but most of these are found pretty similarly in men and women. There might be some associated. For example, there are probably more male former smokers and smoking's associated. But I think overall, there's not much of a sex bias for these different mutations. That may not be true for all of them, but I'm not aware of any. They certainly are occurring in older men and women. Yeah, thanks. All right. Yeah. Right. Yeah, I mean, I think in some ways, evolution, of course, has acted for, when we say reproductive period in humans, we really mean reproduction plus postnatal care, right? So if you died right after childbirth 1,000 years ago, your offspring was unlikely to survive, particularly if you were either the man or the woman, but particularly for the woman. So reproductive period includes that inclusive fitness of being able to care for your children, plus even your grandchildren. So that's so there's going to be selection for our survival and our maintenance during that period where we're likely to be contributing to the passing of our genes on to future generations. In terms of hormonal influences, there have been some studies that have shown this. We really haven't looked at that, but clearly, changing hormones as we get older can definitely influence cancer incidence, but I think it can kind of do it in both directions. So that's a kind of short, quick answer. All right, in the last remaining minutes, I'm probably not going to get into lung stuff, but I do want to talk about this heterogeneity. And I think this is really interesting, again, from sort of an evolutionary perspective, is that we had proposed, and this, again, work that my lab did with Shang Li's lab at USC, that we know that in young stem cell pools, that there should be selection for particular phenotypes, because those are the phenotypes where that stem cell is going to work optimally. But we propose that as we get old, our stem cells are going to become more heterogeneous, because of epigenetic drift, damage accumulation, even damage to the niche. But this greater heterogeneity might allow for selection for adaptive phenotypes that become adaptive as we get older and allow for sort of continued sort of selection within the stem cell pool as we get older. And I'm not just talking about mutational selection. I think this is largely driven by epigenetic changes. And the vast majority of it is probably not transforming. It's probably not oncogenic. It's just the evolution that's happening within us anyway. And so this is work that we've been doing with this team. So all these individuals are in Shang's lab and Marco is in my lab. So what we're doing is we're barcoding these stem cells with lentiviruses that encode little random sequences that can be distinguished upon sequencing of these cells. And then we can either expand these cells and sort them out and analyze their gene expression phenotypes right after culture, or we can put them into a mouse and look at them a couple months later, and that'll become relevant as we come along. And when we looked at these cells at the early time points, we immediately could see that either by looking at intracell entropy, which basically means how consistent are the intergenic association. So if you have this gene expressed at this level and then this gene expressed at that level, how consistent is that across cells? Well, that consistency goes down. It becomes more chaotic at older ages. And you have intercell variability, meaning for any given gene, there's more variability between cells that one has observed in the old, which is the yellow relative to the blue, which is the young. We also see this both within a clone and between clones. And the fact that it's even within a clone that we get more variability in old is interesting, because that says that it's not just that the cells are more variable when we get old, but the rate of generation of that variability becomes increased. What's also interesting is this variability is driven by specific genes. We see specific genes that are more variable. So this is not expression, but variance that we're measuring here as the mice get older. And we look at those variable genes, even when we look at unmanipulated HSCs that never received a barcode, they're also more variable in the old relative to the young. And we further showed that if we just took a random collection of genes, we don't see such variability. So there's something specific. There's certain genes that tend to be more variable than other genes. And when we ask what pathways these are in, I won't get into it, but these are mostly in sort of stress response pathways. So we're seeing increased expression of stress response pathways as being more variable. And we think this may indicate some sort of adaptation that's going on. And so then we did experiment. This is Marco's idea, and it's a really cool one. He basically says, let's take young or old donors, we'll barcode them, and then we'll put them into young or old hosts, but we'll do all combinations. And the cool thing about this, since we can tag these cells and we have their original tags, we can actually follow the same tag from the in vitro culture that we did straight out of the animal to 2 months later after it's been in this animal, and look for the different fates depending on the age of the host that it goes into and other factors like that. And when we do that, for example, each line here represents one clone. And if it's a line that goes across, it's the same clone that went from the same donor in young and went into a young recipient or an old recipient mouse. And you can see that in some cases an HSC is gained, meaning you can see it's in the HSC row here, but not so much here, or it's lost, meaning it was here, but not so much here. So we can look at sort of gains and losses of HSC from the same clone in these different environments. And we can also look at measures of their fitness by their clonal size. And I'll get into this in a second, but I'll just go straight to the sort of the quantified data. What we found was that was really cool is that old donor sister clones have a higher self-renewal in old relative to young host. So basically, these old cells are more likely to have an increase in HSC bias in the old recipients than in the young recipients. And it's the opposite for young. They're more likely to have an increased fitness in the young recipient. So basically, these stem cells are adapted to the age of the host that they're already in. So through a lifetime of being in an old, marching up to being in an old mouse, those stem cells have been adapting to be in an old mouse. And now they're naturally more adaptive into another old mouse when you put them into it. So it basically says there's some sort of epigenetic evolution happening throughout the life of these mice. And we can see this at the same time in terms of clonal fitness. Much greater clonal adaptation in the old going into old than the old going into the young. So again, it's like an adaptation to the old environment. So then we looked at this heterogeneity, and we asked, okay, so let's take the top 30 most successful clones, 30% of those clones, versus the least successful 30% of those clones, and ask, and this is after 2 months in a recipient mouse, let's ask if we go back to that original stem cell before we put it in, that pool of stem cells with that clone, can we see differences in heterogeneity? And indeed, we see greater heterogeneity in the stem cells that ended up being more successful, as if this greater heterogeneity was providing opportunities for adaptation. And then Shang's lab did a machine learning algorithm where they trained the machine learning on the day 0 information that they had, the gene expression, the variability. And they asked, could they predict the success of these clones later on? They also did a late prediction, but I'm not going to get into that. And basically, they got pretty good predictive value, from 60 to 80% predictive value when they looked at stem cell self-renewal or clonal fitness index. So they could basically predict how successful an HSC would be based on its behavior before transplantation. And they found that this algorithm, this machine learning, was driven by particular genes. And when they asked, were those particular genes prognostic in leukemia, they indeed were. So more expression of these genes driving this fitness phenotype was associated with worse outcomes for a patient that had AML. And I'm going to step down here so I can actually read this. So what I've shown you is that intrinsic aging basically encodes for variable clonal starting positions that interact with the niche age to shape hematopoietic outcome. And that old HSCs exhibit greater heterogeneity and rates of generation of heterogeneity driven by specific gene sets. And old HSCs exhibit higher self-renewal and fitness in old relative to young host, likely reflecting an adaptation over life to the aged altered niche. The pre-existing heterogeneity predicts both higher clonal fitness and higher self-renewal in the old host after transplantation, which provides greater phenotypic variability upon which adaptation can happen. And finally, that a machine learning model can predict HSC clonal fate, and these key variable genes underlying this model are predictive of survival for AML. And I think the question is aging-related adaptation to the changing niche, is it good or bad for us? And I think the answer is yes. It's probably both. So in some ways, this adaptation might help better maintain hematopoiesis as we get older. But we can also imagine that some of these adapted clones may be better substrates for future transformation. So I think we need to do more studies to determine what the answer to that is. So I'm going to show you 2 seconds of this. No, I'm not. I'm just going to skip through it. So this is all the studies of clonal mutations in the lung. A lot of it was done by Ed, postdoc in my lab. But I'm going to skip through it. Basically, suffice it to say, our tissue of lung becomes loaded with mutations as we get older. They're expanding. They're being selected for. We can show selection. But and it's smoking dependent, but not exclusively smoking dependent. But what's kind of cool is that many of these mutations are not the ones found in cancers. In fact, we find that a lot of the expansions, there's no association between how frequent they are in cancer as to how frequent they are in our normal tissues. And we did computational modeling with my colleague Elan, and we modeled these onto the structure of this gene, PIK3CA. We find that a lot of these blue mutations found in normal tissue, expanding in normal tissue, are at this interface with this negative regulator, along with the tumor mutations, which are in red. And just like the tumor mutations, they tend to disrupt that interaction based on sort of biophysical mechanisms. And in some ways, I show this because, again, this is something I could not have done, my lab could not have done, but we had a good colleague that could help us with it. So if you want to ask an important question, you have to find the right people that can help you do it, because you really need to think big. And thinking big often means going outside your own area of expertise. In fact, of everything I've shown you today, I probably could myself do about 5% of it, maybe. So I'll skip over this, but basically to kind of sum up is that we think as we go through life, aging, exposures, lifestyles, that this is affecting our stem cells, probably mostly through epigenetic changes, reducing fitness, increasing heterogeneity, and along with damage to the niche and altered selective pressures, this is going to increase selection for mutations that are adaptive to this changed environment. And this can lead to this massive expansion of these different clones in our tissues as we get older. But this isn't just about cancer, because these clonal expansions, they're not just fodder for evolution to malignancy, but we think that they can reduce tissue function. And we're getting some data that suggests this as well, that could lead to the risk of disease and aging. And of course, there's interplay, because this declining tissue function could further promote malignancy development. And I'll skip over this, but go to the thank yous. So my lab been fantastic. And I think I thanked people along the way that were involved in this. We have great funding. And I'll also mention that if anyone's interested in aging and cancer, please submit to our journal. And if you want a page charge waiver, contact me. And as I answer questions, I'll go back to this slide just so it's up there. All right. Thank you so much. Do we want to have the, there we go, I just turned it on.
Right. Any questions?
There's one back there. I suggest that people on complete opposite ends of the room ask questions. That way Dr. Bill gets his exercise.
Oh, it should be on.
It should be on.
Hello?
Yeah.
Oh, there we go. So as humans have had an average of living longer, these mutations have arisen. So right now, the average age of living is about 75. If we bump that to 85, can we expect to see more mutations and therefore more disease, more cancer? Am I understanding that correctly?
Yeah, well, it could be, but it also, I mean, I think that one thing I didn't get to so much in the last part was that smoking had a big influence on these patterns, right? So, I think if we all started living much basically healthier lives, we might keep the more damaging mutations at bay. So I think that there's a lot of clonal evolution that's happening then to us. Even if you live the perfect life, you're still going to have a lot of this clonal evolution. It does create some risk, but I think we can modify which clones are expanding. And as I tried to show you with the clonal metapoiesis studies, not all clones are equal, right? Some clones are much more basically damaging and dangerous than other clones. And so I think if someone who lives longer probably will have more clones in their body, but hopefully if they've been living sort of not smoking, eating well, exercising, hopefully they're not going to have the clones that are more likely to contribute to, for example, tissue decline or cancer. But inevitably, this is probably going to always be a limiting factor for human lifespan.
Okay. Thank you.
Sure.
Continuing that last question, if humans did continue to live longer, is there a chance that selective pressure would adapt and find a different way to a solution to dealing with these cancers?
It's definitely possible. I mean, let's say that even as sort of medically, we could figure out that, okay, if a certain diet, a certain intervention would direct these clones towards less malignant, and in this case, I mean malignant, not just in causing cancer, but basically causing reduction in our fitness. So if it could direct it to more benign types of clones, then yeah, we might be able to push out our lifespan. Now, I'm not saying that these clonal expansions are the only reason we die. So how that's going to affect our overall survival, I think, remains to be determined. But because, obviously there are other reasons that our systems fail. And you're only as fit as your weakest link, right? So if your heart goes and your liver was fine, well, your heart still went. Yeah.
I have a question. So could you apply the same method if you live in like an urban area and rural area?
Oh, yeah. So I was involved in a study that was out of Charlie Swanton's lab in England. And so we actually did the same sequencing methods in the lung for that study. And they were looking at people that had different exposures to pollution. And basically the conclusion of the study was that that there was selection for mutations, for example, in EGFR and in KRAS that was proportional to how much pollution exposure someone had. We didn't get that data directly from the humans, but we extrapolated it by also doing mouse studies. But so yes, you could imagine that just like we did smokers versus non-smokers, if we did a study where we compared people based on how much pollution exposure they had, which I assume is kind of what you meant by urban versus rural, but maybe not. But
Yes.
You might expect to see similar dependencies in terms of certain mutations being expanded out dependent on pollution exposure. And my colleague Amelia Lim up at British Columbia, she's actually doing some very similar studies that will directly answer that question. It's a bit complicated because we often think of rural as pristine, but people in rural areas can get a lot of exposure to pollutants too. I mean, farming can be associated with a lot of particulate exposure. So I don't know if it's going to be a clean study from that perspective.
Okay. Thank you.
Yeah. Any other questions?
I just wondered, women are reaching estrus earlier.
Mm.
I don't know what that would do to the things that you're.
It's definitely probably has a role in the incidence of breast cancer. So, I mean, as you probably know, women reach estrus at 11, 12 years old now. And historically, meaning hundreds, if not thousands of years ago, it's probably closer to 18 or 19. And the other thing is that women in primordial times, I don't know if primordial is the right word, but ancient times, basically, would get pregnant soon after, and then they would basically nurse that baby for 3 years. So there's a large reduction in the number of menses. A woman in ancient time went through far fewer menses in her life than a woman in modern times. That's been given us one explanation for this increase in breast cancer. I don't know if it's the only explanation, but I do think that we need to consider that the modern way of childbirth, meaning that with birth control, now more women in the United States have their first baby after 30 than before. So that's really a milestone. And later childbirth is associated with higher risk of breast cancer. Breastfeeding is associated with reductions in breast cancer. So I don't know if we know the explanation for the why behind that, but it sort of says that we're living under conditions now that are disconnected from what our genes evolved under. So that's sort of a long-winded explanation for that. But yes, I definitely think that earlier menses, it may be contributing to, if you look at breast cancer incidence, it's interesting. Most breast cancers follow that same curve I showed for colon cancer, leukemias, and so forth. But there's a little hump off to the left where women at earlier ages, like in their 40s, are getting breast cancer. And some of that is actually pregnancy associated. So it's postpartum breast cancers. And there's some explanations for that. There's some researchers at my institution that are studying that. But that may be more of what is being influenced by modern conditions.
So would you say besides preventing somatic mutation, stabilizing epigenetic changes would be also a thing we should consider for our better chance to not get disease or cancer?
Yeah, I would say that doing either is be extremely difficult. I mean, our mutation rate is already super low. Making it lower would be very difficult. I mean, obviously we can avoid mutagens, right? Don't smoke, don't get exposed to no mutagens. But I think that almost the generation of diversity, that's going to happen. And you could make it worse by doing things, but that's going to happen independent. I think what we could do is we could find ways of modulating tissue environments that reduce the evolution of these clones. Because it's not just about the diversity that's generated, it's about the selective pressures, as we talked about earlier, that would drive particular phenotypes to be adaptive. That, I think, we have more control over.
All right. So let's thank Dr. DeGregori again. And before anybody gets up, we'll do a couple raffles. Everybody got a ticket. So if you want to get your tickets out. Danielle gave us the gold basket of fun. That would be fantastic. Actually,
I older than you do.
no,
Hold on.
just give me a ticket. No. And then if you want to come up, if you get a ticket, we're giving away some t-shirts. So you can come up here. If you want extra credit, we have the sign-up sheets and we'll have those outside in just a second. All right. So the first ticket, 275266. Everybody's running away. Just give me another one. All right, 275261. Yay! I'll take it. Oh, that's yours. Oh, call it. Okay. I was supposed to come down. Yeah. All right, 275263. You sure you're shaking it up a little? He's good at
Yeah,
grabbing.
that's copy a little bit.
I don't know. 275238. Go ahead. Sit there and ask Christmas.
Oh, that's good.
All right, then we have 275286.
One more.
Sure. 275272. Oh, yeah. Okay, one more. One more. Yeah. And 275264. That's
You can-
so. Just go ahead and sit there. We'll get